Hi guys, welcome if you are joining me. I thought I would do a bit of some questions, and if you have any questions then please do feel free to ask away. But let's have a look at setting a assignment today. So this is aiqchat.com. If you haven't seen the revision platform, please do check it out. Let's see what questions it's going to give us today. So the first thing it's going to ask us is, what enzyme is used in the polymerase chain reaction to make copies of a piece of DNA? So which of these is the right answer? So whenever you do this, make sure you are thinking through the topic area and not just going straight to guessing the answer, because that's not going to help you when it comes to the multiple choice questions. So which enzyme is used in a polymerase chain reaction to make a copy of a piece of DNA? So we have got DNA polymerase 1, transferase, polymerase alpha, or reverse transcriptase. So in this case, I know with polymerase chain reaction, I need DNA polymerase, and it's normally Taq polymerase. And the Taq polymerase is used because it is going to withstand the high temperatures. Hi Christine, I finished the first few chapters of A2. Can I finish by late April effectively? Yes, so I would say when it comes to the year 13 content, I would normally, when I was in the schools actually teaching it officially, I would be aiming to finish by the end of April, if not the beginning of May, depending on if we've got extra exams, if we need to catch up on PAGs, things like that. So that's what I would be doing. Hi, Daryl, how are you? Guess what? I am currently building what am I doing? Biological molecules and membrane study guide at the moment. Not just that, it's also going to have the immune response in there as well. So that's what I'm currently working on. I was doing so much of that today. But then I've been tutoring all evening, and so now I am doing some questions. I thought I'd jump on here and answer any questions for any students who might want a bit of help. And we're looking at AIQ chat. I started quite late for content and quite worried about not finishing time, as I effectively I've got 2 months. I wouldn't worry too much about that. So what I would say is it's difficult when you start late, but there's still time to get ahead. The one thing I would say is make sure you're not revising from start to finish, thinking that you're going to get all that content done. What you should be doing is using the time to focus on what you don't know and what you find difficult. Yes, there's only 2 months left, but actually you've still got time. Hi, Chidima. It's been a long time since you have been on here. But I would definitely say you've got time to do it. It's just about focusing the time so that you are able to figure out what you don't know and work on that. If you haven't done, checking out AIQ chat, I'm obviously bigging it up because it is my revision platform, but that is built to help you and guide you and allow you to learn things in a much faster way, because you're chatting back and forth. If you use it properly, you're chatting back and forth with the tutor, and so that way you are assessing your knowledge as you go. Yes, unfortunately, Chidama, it is only module 2 for free. If you want access to the other modules, then you do need to pay, but we did reduce the cost down to £5.99 for the whole month, and so therefore I would say it's not that expensive. I mean, it costs us, it's currently actually costing us more money based on insurance and the platform and building all the other things. So yeah, £5.99 is how much it costs for you, which is a reduced cost, and it shouldn't, I mean, it's what, one coffee, and if you want half a biscuit. I currently use your videos and flashcards. I'm an independent candidate with an offer of ABB. So, okay, so if you're currently using my videos, that's great. Have you checked out my revision study guides that I've got on my website? If not, then do check them out, because what I've tried to do is consolidate the content also to a high enough level to make it understandable. And if you buy them in bulk, you actually get like £68 off. So it's better to buy them in bulk now than just buying one or the other, because you actually get a whole load more. Also wanted to ask, the solute concentration increases as you go down the medulla. What are you talking about here? The solute concentration. Are you talking about the medulla in the kidneys? So you say medulla and I'm like, talking about the medulla in the adrenal gland? Are you talking about the medulla in the kidneys? Are you talking about the medulla oblongata? I'm assuming you're talking about the medulla in the kidneys. Okay, so you wanted to ask, the solute concentration increases as you go down. So as you're going down the descending loop of Henle, what you have to remember is, as you go down the descending loop of Henle, the water is going to leave the descending loop of Henle because it's permeable to water, because the water is going to leave by osmosis from an area of high concentration to an area of lower concentration. So as the water leaves, that is going to result in a increase in that solute concentration because you've got less water there. So that would be where I would say that hopefully answers your question. Do you think knowing all the content would be enough to get at least a B? No, I don't think knowing all the content would get you at least a B. I think knowing the content and solely knowing the content will get you to about a C grade. What gets you to the B, to the A star is how you are able to take that knowledge and apply it to the question that's in front of you. That's going to be the difference. So I would definitely say it's about the application. So if you can, try now to do as many past paper questions as you possibly can, because they will be about pushing you to apply the knowledge that you have. You've got a test on communicable diseases next week. What might be the hardest question? Most students struggle with the communicable diseases part of it, with actually talking about the difference between the B cells and the T cells, and understanding clonal expansion and clonal selection. So do you know, for example, what the different types of antibodies are and how they are useful? Do you know that it's the B cells that are going to be selected for, and that actually you need the B cells and the T cells? So the B cell needs to be activated along with the cytokinins that are coming from, or interleukins that are coming from the T helper cells before it will fully activate. So you need clonal selection to happen with regards to the correct antibody, or the correct B cell with the antibody on its surface being able to bind with the antigen, along with the T cell actually encountering a antigen presenting cell, and then releasing the interleukins, which will then fully activate for the B cell to go through clonal expansion. So clonal selection is needed, and clonal expansion, and linking that to your T cells is probably where I say most students seem to struggle. So Chidama, but why, for example, is the upper part of medulla less concentrated than the lower part? Well, as you're going down the medulla, you're talking about the concentration difference between the concentration of ions, concentration of urea, concentration of water. So it's all to do with where the water can go. If the water can leave from the descending limb, it is therefore going to become more concentrated for the ions inside, because they're less dilute. There is more of the ions with regards to the volume and concentration of water. if that makes sense. Can you tell I'm a bit tired tonight? My eldest is not having the greatest sleeping at the moment and is keeping me awake. Kicked me out of bed. 5 a.m. I'd had enough and I was in her bed. Ray, can I see your message? Do you know what, Ray? I saw, potentially, I can... no, I've only had one person do a smiley face on the YouTube. So if you're on YouTube, I don't know what is going on. I've only got one smiley face, so I can't see any messages there. Have you been putting messages on my videos? Because I just noticed as I logged in that I'd actually missed some notifications that hadn't come through. So if you have been writing comments on my videos, I apologize. Sometimes it doesn't come through to me, and so it takes me a while to see them. When I do, I will reply. And then at the ascending limb, how come some sodium gets actively transported out and some diffuses out? So it's all to do with the active transport is about moving it from a low concentration to a high concentration. And so therefore you're using the pump. With regards to some of it diffusing out, it depends on the concentration gradient and whether there are the proteins allowing for them to move through. Do you have any tips on how to structure different types of questions? When you say... No, the YouTube Live, I think. Yeah, the YouTube Live doesn't seem to be working very well for the chat. I don't know what is going on. It literally has. I always find it bizarre, which is why I put TikTok on at the same time. It's given me one person who's chatting, and that is it. No other people are chatting on here. So I've got Matty UX Vibe. Oh, there's 5 of you watching. Wow. Thanks, guys. Yeah, so I don't know why. I don't think you answered. No, Chidima, I'm kind of jumping between the two. Give me a second. I'll come back to your question. One day you'll understand the kidney. Have you looked at my excretion workbook? But if you think about it, eventually the sodium will be at equilibrium, so it can no longer leave the limb, so it requires active transport. Yeah, so it's... The key thing is to know, whenever it's active transport or whenever it is facilitated diffusion, you're looking at the movement of the ions from one place to another. Are they going down the concentration gradient or are they going against it? And so if you've got channels that are allowing it to move through, it will diffuse. If you have got the... what am I trying to say? The pumps, it has ATP that will allow it to move against the concentration gradient. I just don't understand how. What do you mean you don't understand how? The bottom part of the medulla is saltier than the upper part because does water not move out at all? So the water can only move where it's permeable, so it can only move from the descending limb. It doesn't move out of the ascending limb. When we look at the limb, the top half has less channel proteins compared to carriers. Yes, it's all to do with difference in the limbs and the proteins that are present. Absolutely. So we say that there is the permeability in the ascending limb. That permeability is obviously to the salts, but not the water. And the descending limb is about the water. So because they work against each other, that you get this counter current. So as it's coming down, the descending limb is going to let the water out. The ascending limb is pumping the sodium through, and whenever you have sodium move, chloride kind of follows with it and it goes down its concentration gradient. Now be careful, Ray, always say either down the concentration gradient or against the concentration gradient, depending on what it is that you are trying to describe. If you say it's going across, you won't get the marking point for that one. So it's those key words that are going to make the difference of you being able to get marks in an exam and or not. Sometimes as simple as something like that, that is a no-go. Never say across. Down or against are your key words. You are very welcome. Thank you for calling me your queen. I'm not so sure I'm a queen today. My hair is a bit of a mess. Got my plaits in. I'm very tired. Very, very tired today. But as I do, here I am on a Wednesday. Oh, it was very exciting actually. Today was my first day that I actually got paid on YouTube. I'm so excited. I mean, £76 is nothing to like knock, because actually £76 is 6 months of me being able to be paid, as in being able to earn from the you guys watch my videos. Thank you so much to you for watching my videos. So you watch my videos, and that therefore means that sometimes they drop in some ads there, and if you watch the ads, even if you skip the ads, because you've watched my video and they've managed to put a bit of advertisement in there, I get like a little bit of that income. And so yeah, so I obviously built the YouTube channel and I started being able to earn in September. So since September till now, I've managed for the first time to earn on my YouTube channel. So I'm super excited about that. Yeah, it's interesting how different it feels actually saying that, oh my gosh, I've actually managed to earn some money on my channel. For example, with explain, describe, and compare questions, how would you structure? Right. Okay. Thank you for that. That's a really good question. So for OCR A, when they say explain or describe, so describe is say what you see or say what you would see. So a describe question could be describe the data in the table or the graph and then explain what that means. So for example, with a describe question, it could be temperature and it could be enzyme activity. So if I increase the temperature, what I would see is an increase in the rate of the reaction up to a point which it would then reach its optimum, it peaks, and then it would decrease after it had gone past its optimum. So that's the describe. So I'm saying what I can see in the graph, or I'm saying what I would see in the biological process, whatever it is. When they then want you to explain what's the science behind that. So if I am talking about temperature and I'm talking about the enzyme activity, because that's what I decided that was what I was going to put today. If that's the case, I've just got a flower. Thank you for the flower. What I would say is for that type of question, explain as the temperature is increasing, there is more thermal energy, therefore more kinetic energy, therefore there would be more successful collisions. Those successful collisions would result in a higher rate of reaction up until the point at which it reaches its optimum. After its optimum, then what you get is the increase in kinetic energy is actually going to cause more vibrations within the enzyme, the protein, and that will therefore affect the tertiary structure, and that will cause it to denature. So therefore the describe was increase in temperature meant increase in rate of reaction up to a point where it peaks, and then it decreased because I went too high. The explaining was giving the science behind it. So I think in a very logical order. Whenever I try and give answers, I always try and go back to the very basics and then build from them. A lot of students jump to the end point and they miss some key marks because they think that what they're saying isn't going to be relevant, or they're saying that you are... I forgot what I was saying because I just saw someone else's message. So, but you get what I'm saying. So in a sense of, you jump to the end, you don't answer half the question, or you think that what you're trying to say is too simple and it's not a marking point, but it will be. We'll be watching those ads in Phil now. Thank you. I don't know if it makes a difference, but thank you if you do. Can you explain a flow chart without specifics of how an antigen in the air causes infection? Right. Okay. Now, what do you mean? Do you pay for AIQ chat? If you pay for AIQ chat, it is £5.99 a month. But you get 7 days free trial. So if you want to go and give it a shot, play with it for 7 days. See if you like it. If you don't like it, cancel the subscription. And you won't be charged. What I would say is the best thing you can get out of using AIQ chat is by chatting when you get it wrong. I would expect you to get maybe 1 or 2 wrong. At the start, you'll get quite a lot wrong if you don't know your content, but you'll quite quickly pick up on how to get better at that. And that's the whole point. It's about repetition, repetition, repetition. The more you repeat it, the more you start to see the right answers, the more you're able to retain the information, and you don't feel like you're learning. You are engaging. And also what it does is it trains you to read the question, because some of the questions are very similar and I've just adapted them slightly, and so that therefore means you have to read the question. You think it's the exact same question, but it's not. And so if you're just thinking, oh, I know this because that's the answer it was before, that you'll catch yourself out on that one. But what you gain from using it is actually chatting with the AI tutor, because that is being built to ask you questions. It's been built to guide you to understand why the right answer is the right answer, rather than you memorize, oh, that was the right answer, and moving there. Right, flowchart. Darryl, I am going to do this for you. Give me 1 second. I'm going to do it on my board for you. Let me just hide that window. And let me bring up this window now. Right, so you want a flowchart of how an antigen in the air is going to cause an infection. So when we have something which is foreign, it is going to have the antigen on it that is going to be recognized as being not the self. So that's the key part. We're just going to give some keywords to go through. Right, so we've got an antigen that's foreign. It's a not self. It is a protein, which is potentially, it's a protein, potentially it is a polysaccharide. It depends what it is, but it's an antigen, is recognized as not being self. What's going to happen is you're either going to have your phagocytes do phagocytosis, and those phagocytes, when they do phagocytosis, they're going to engulf. Hold on, I need to move my keyboard out of the way as I write my board. They're going to engulf. My writing is getting so bad. Phagocytosis. They're going to engulf and digest. Now, some students will be like, oh, that's enough. Now that's not enough. Okay, that's a GCSE level. Phagocytosis, you learn at GCSE. Engulf and digest, you learn at GCSE. So phagocytosis, I would be saying my keywords are phagosome, lysosome, lysosome, and lysozymes. So what I'm doing is I'm now getting my flashcard keywords. Phagocytosis, if I'm going to do phagocytosis, I need to talk about engulfing into a phagosome. Lysosomes containing lysozymes, which are then going to fuse with my phagosome. I've then got my phagolysosome. I'm going to digest, and that is going to break down the pathogen or the antigen. And what's going to happen is I'm then going to have a way of presenting the antigen on my surface of the cell. So I've got an antigen presenting cell. So we've had an antigen, we've had our non-specific immune response, our phagocytes have encountered them, they engulf and digest, they present it on the surface. So here's my phagocyte, which is now going to present that antigen on the surface of its cell. It's now an antigen presenting cell. Presenting cell, because it has a MHC complex. Okay, so now what happens is that antigen presenting cell is now going to encounter a T cell, and that T cell is going to recognize that MHC complex. It's going to recognize this antigen presenting cell is going to be recognized. I'm not really doing this as a flow chart. I kind of am, but I'm not. You said without specifics, but I'm not going to do it without specifics because I can't. And this is a T helper cell. So once the T helper cell is going to encounter this antigen presenting cell, that is then going to send interleukins. So these interleukins are then going to be signaling for more phagocytes. These interleukins are going to be going and activating the B cells. So they activate the B cells to go through clonal expansion. So my B cell, the correct B cell, is going to need to encounter the antigen as well. And that encountering the correct antigen is clonal selection. So we need clonal selection, and it takes time. It does take time for the clonal selection to occur. So we need clonal selection. We need the B cell to be activated, clonal expansion, and then the B cell will go through division and it will then differentiate so that we end up with, I've got no space on my board. Give me a second. Let me move on to the next one. So then what we've got is our B cell, which is going to then go through this expansion, and you've got your plasma cells, and you've got your memory cells, and then you've got the antibodies. So you said, can I go through a flow chart of the antigen in the air causing an infection? So the antigen in the air is going to cause an infection. Is not what I answered. I actually answered, how are we going to fight against it? See, this is the thing. I've read your question, but I've gone a completely different tangent and spoke about something that's not actually what you asked. So I now need to go, hold on a second. What did the question actually ask me? How an antigen in the air causes an infection. So it causes an infection depending on what it does. The cytokines, what they do is when they are released by the T helper cells, they will cause inflammation of the area. So they'll cause inflammation of the area, which is they will cause the let me think, what am I trying to say? Endothelial cells to become leaky. And that allows the phagocytes to get into the area. It floods the area with liquid, and that therefore keeps it there for the phagocytes to get to. But also by flooding the area with the liquid, what we then end up with, so if this is my tissue, there's my cells, very basic by the way, and there's my blood vessel. If I am causing more leakage into for more tissue fluids, see, I'm going in a completely different avenue now. What that's then going to do is that's going to flood the area, and that will then get drained into my lymphatic system. And if it's drained into my lymphatic system, that therefore is going to be circulating round in the lymphatic system and encountering more of my phagocytes and also my T cells and my B cells. Does that answer any of the question that you asked, Daryl? As in primary defense and secondary specific, blah, 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 blah. Yeah, Ray, the module 2 is free. All of it is free, and you get 5 chats as well for free. What's an MHC complex? So an MHC complex is basically it's like a specific part on the surface of the cell, which is a complex that is on the cell, and depending on the type of cell you've got, you don't need to go into this detail, but you have MHC complexes, which is MHC1 and MHC2, and different cells have these different complexes. And so it depends on the type of cell that they are, it depends on what they need to do. That's going into more detail than you need to know. You just need to know that it becomes an antigen presenting cell. That would be my advice on that one. Would it be helpful to create my own personalized structures for each type of question and word banks? If you have the time, then I would say yes, but it depends how much time you've got and whether it's a good use of your time, would be my answer to that question. Oh, you're very welcome. I'm glad it's useful. This is the thing. Sometimes going off in tangents does actually help. And sometimes it doesn't. It's a funny one. Right, let's have a look. You're currently in year 12. Oh, okay. If you're in year 12, you have got plenty of time. Absolutely 100%. Turn everything into your own work. I'm not saying don't buy flashcards. I'm not saying don't buy study guides, as in mine. What I'm saying to you is you need to actively be doing the work. You need to actively be thinking about the content. You need to actively be going through the processes so that you understand what it is that you're actually being asked. And you can also then talk about with other people as well. Like if you use like a study buddy or anything like that, that will help you to understand what you do know and what you don't know. And you can then use the time to improve your knowledge. So yeah, 100%. If you're year 12, you've got plenty of time. Do we need to know about page 53 gene? Oh, page 53. P53 gene for the G1S checkpoints. No, you don't need to know specifics about the checkpoints. You need to know the checkpoints are there. You need to know that the checkpoints are used to ensure that the cell is the right size, the DNA hasn't been replicated in the wrong way, or that the DNA doesn't have damage before going through into S phase. But no, you don't need to know specifics. If they're going to give you a question about those sorts of specifics, let's try that again. You will be given the information in the stem of the question. Hold on. YouTube deleted your comments. See, this is what's really bizarre. I've not got any comments on the YouTube channel, which is why I'm kind of using you guys here rather than the video. So I would say, yes, it should be a combination. Right now, you've got plenty of time. Using a combination of all the different things will help you massively, because if you listen or watch a YouTube channel, and obviously mine is the best, if you watch the channel, if you watch the video and you still don't get it, go and use the textbook, go and use the study guides to see if that then helps you to then formulate your own answer. There's lots of different ways in which you can learn. If you are someone who is more visual, I'm more visual. I need to see things, which is why I need to draw it down, which is why I have built my channel and my study guides around that visual graphic side of things. Is why I've learned Adobe Illustrator, so I can make my own graphics for you, because I am a visual learner. I can't read textbook like word, there's just too many words for me in the textbook, and it takes a long time for me to retain it. I have to read it again. So it really just depends who you are, how you work, and what's best for you. Just because something works for someone else doesn't mean it's the right thing for you. So figure out, are you someone who does bullet points? Are you someone who does mind map diagrams? Are you someone who prefers to have post-it notes in lots of different places? Just figure out what works for you, but don't waste your time on something that feels like you're doing lots of work. And reading that textbook is a good use of your time if you're gaining knowledge from it. But if you're just reading it and none of it's going in, that's not a good use of your time. Yes, you'll have spent hours studying it and reading it, but that's not going to help you to then put it down in the questions in the exam. For OCR A A-level, do I need to evidence for semiconductor nitrogen. So interestingly, they did a question on the heavy isotope and the light isotope for semi-conservative replication. So they could give you questions on that, but that doesn't necessarily mean that you need to memorize it. But it is important you understand about the different isotopes that you can have and how that's useful in being able to understand the biological processes. So the Michelin style investigation is a key one for proving the theory, giving the evidence for the theory of semi-conservative replication. And this is the thing about science. We use science to do all these investigations so we can find out about what's going on, and we can come up with hypotheses, and we can either prove those hypotheses or we can disprove them. And it's about understanding that with the advancement of science, we learn more and we learn new things, and we change our assumptions based on the evidence that's out there. So answer that question. You don't have to memorize it. You will have to be able to interpret it if they give you a question on that. What's the best way to revise? I don't know, to be honest. I find that I have to do a lot of repetition. So I do a lot of work in the background, and I have retained a lot of information, and that is hopefully evident to you guys that I know my stuff, but there's still things that I forget. So I have my little book. I have about 5 of these. I have a book, and what I do is I'm constantly writing little thoughts down at the time when I'm making questions, I'm thinking about it, what am I doing? I'm constantly writing notes down. I'm not doing it in a very methodical way as what some people would do. I used to think I needed it to be very specific, methodical, and now I know that's not how my brain works. My brain is just a big massive mesh of mess. But I'll pick up a book and I'll open a page and I'm like, oh yeah, I remember, I was reading about that. Straight away I've got it and I remember it, but I'd forgotten it. So whenever I come up with these things, I write it down in a notebook. And so I've got like this one, I've got 5 of them, and I've also now got like really little ones that can fit in my bag, so that if a thought or something comes up when I am randomly waiting on the girls at pickup or whatever, I can just quickly jot it down. So it really just depends who you are. I do things through drawing. I do things through writing it down. I try and abbreviate a lot and limit my I can't be bothered to write it all down. And 2, your brain can't retain lots of information, but you can do things like chunking things together. So if I can chunk things together, or if I can come up with something that makes sense to me, then, and helps me to remember it, then that is what I will then grab onto, and I will use that. So the alphabet, for some reason, I love using the alphabet. So whenever you listen to my videos, you hear me talk about, whether it's the left atrium or the left ventricle, what does that link to? The aorta. How do I know? Well, because it's on the left side, so it's the letters A to L. And so therefore I've got my bicuspid valve and I have my aortic valve, and the only one that doesn't follow that rule is the pulmonary vein, which goes into the left side. So I have the alphabet as my way of remembering. When I'm doing the sodium potassium pump, I was doing that earlier today, and when I do that, I talk about the fact that Na plus, when I'm writing a sodium ion, I'm writing Na plus. Na plus is Na plus, OUT, out, 3 sodium are going to be pumped out, K plus potassium, 2 potassium in IN. So it's about finding what works for you and basically going down that route of, if it works for you, use it, find ways, but be careful, because sometimes you can end up with misconceptions. And if you've got a misconception and you just run with that misconception, when it comes to your exam, you'll write about that misconception. And so therefore you need to make sure that what you're writing down, what you're doing, whatever revision style you're doing, it is accurate what you're doing. Sometimes I remember things because I know the answer is the opposite of the apparent. Like, you might assume it's from X to Y, but in reality it's Y to X. It is funny with the things that you get to work for you. My brain is an absolute mess. It is such a mess. But now I understand that, and now I understand that I go in tangents around. It means that I am way more productive, that I'm able to retain information more because I understand that my brain doesn't go down from A to B. I will go from A to F to G to C to A to B to C again, but I'll still get to the end point. I just have to know that although my brain is a mess, when it comes to writing it down on the paper and answering the question, I have to take the time to read the question, to formulate my answer, to then get it down so that I can make sure I don't write the wrong answers, if that makes sense. Do you know what the other thing is really fascinating? If you want to know. So for those of you who don't know, I have ADHD, which means that I obviously have got lots of racing thoughts going on. My brain is working really, really quickly. But the funny thing is that I'm talking right now, I can't remember half the things I say. So when I do a lesson, every lesson, yes, there'll be things that I will repeat and repeat and repeat because they're necessary, like key words. And whenever I do that, it's important. But there'll be times when I'm saying things and I'll be like, I have no recollection what I've just said. None whatsoever. And so therefore I'll be like, okay, so now we're going to go and talk about something else. And so the lesson will be slightly different because I'm coming at it from a different angle. It's really interesting reflecting on my whole 20 years of teaching and how it has got to the point where I am now doing this. This isn't planned. This is literally me sitting here answering questions as you go. I'm doing biology paper 2 mock tomorrow. Is there any topics you remember? Yes, there are some topic areas that I remember. Am I going to help you out with those? What would I suggest you do? So you're doing a paper 2, which means it's modules 4 and 6. So I would say biodiversity would probably be one I'd be looking at. But again, it depends on what your teachers have done, if they've given you the full paper or whether they are adapting it. If they're anything like I would do, I would be changing it slightly to make sure I can see exactly what level you're at, rather than me just giving you the paper that you may have heard from someone else about what's in it. I would say, I'm trying so hard not to give away. There's a big massive 6 marker and I know exactly what it is and I'm trying so hard not to tell you. Basically, I would say definitely make sure you are confident with your genetics. But when I say genetics, be aware that, remember, it's module 4 and module 6, which therefore means it's classification and evolution, as well as biodiversity, as well as communicable diseases, alongside patterns of inheritance, alongside ecosystems, alongside manipulating genomes, things like cloning. If you look at, interesting, if you really want to know what I think will come up in your module 2 paper, or your paper 2, you should watch my prediction for the paper that I did just before the actual exam. So I would have done a live on the 20, oh gosh, so it was June the 11th, I want to say, that I did it, potentially. Would that be right? No, it must have been before that. Maybe the 6th. But yeah, I did some predictions there and everyone came out saying I predicted pretty well. Darrow, there isn't one topic that's most application heavy. They're all application heavy because biology is the bigger picture. Biology is so interlinked that you can apply the content to any situation. Obviously, enzymes comes up huge amount because enzymes are everywhere. Biological membranes and transcription translation are very much application based, because it's like is there a mutation? What causes the mutation? What if it's a silent mutation? But that's year 13, and I believe, Dara, correct me if I'm wrong, you're year 12, so you've still got that to come. So yeah, I'm trying to think what else there is. Yeah, I thought you were year 12. Interesting. Year 12. No, that would have been paper 1. Actually, you've already done paper 1. I think the fact that the fish and insects, etc., came up hugely in paper 1 makes me think it's not going to come up in paper 1 for you this year. If they do anything related to that topic area, I feel like it'll be a practical on diffusion, maybe, and surface area to volume ratio, like actually practically based rather than the content. And the other thing that's been really fascinating is how much they have taken the content and given the content to you in the question, and then really thrown you a curveball by saying, now use this to explain what's going on in this practical, or use this to evaluate these scientists' conclusions. Would you recommend writing notes for each bag? No. Dara, I would not recommend writing notes for each bag. That sounds ridiculous. Would I recommend writing up some key things from your practicals? Yes. And what I mean by that is any practical that you did. So if you've done, for example, the calibration curve, so quantitative measurements, then yes, writing notes about what did you do, the fact that you had to do a zero dilution, the fact that you had to zero the colorimeter, the fact that you had to use a specific filter, those sorts of things. I'd say, yeah, absolutely 100% write notes about that. If you're drawing graphs, what were your key things about the graph? I mean, graph drawing is always going to be one that they're going to assess. I feel like they didn't do the histogram this year, which I thought they would do. So 2025 they didn't do histogram, which makes me think a histogram is coming up 2026, so this year, because we're in 2026 now. That would be a little assumption that I would make. But yeah, it's hard to know where they're going to go. It's hard to know what they're going to do, because obviously they make these papers up and they can't use papers or questions that they've used before. They might use same diagrams. I've seen them use the likes of a graph again, or the I'm trying to think, this my brain is completely shutting down. So using things like graphs, using things like tables, using things like key bits that won't change again and again, but changing the emphasis on the question. So for example, because you've done paper 1, I would say they looked at synapses a very small amount in paper 1. So I feel like neuronal communication will come up a bit more this year, but maybe not the synaptic part of it, maybe the action potential, because they haven't done very much on the action potential. But I might be wrong. Who knows? And this is the key thing. You can't just put all your eggs in the basket that I might predict it right, because I might predict it wrong. And so I would say make sure that whenever you are doing your revision, that you have a solid knowledge of most of the topic areas. Module 2, 100%, you should be absolutely nailed down, got that 100%, because biology is module 2 in application. So if you're looking at biological membranes, if you're looking at nucleic acid and nucleotides, if you're looking at biological molecules, all of that comes into context when we say, oh, respiration or excretion or neuronal communication. That is biological membranes. That is transport across the membrane when you look at the action potential and the depolarization, repolarization. So module 2, So if you think about phagocytosis, if we go back to your phagocytosis, or we go back to your plasma B cells, protein synthesis is huge. So nucleic acid nucleotides is massive. And what else would come up? So nucleic acid nucleotides is huge. Cell differentiation, diversity, so 2.1.6, because you need mitotic division to occur so that you can go from having your cell into clonal expansion. So mitosis. Let me think. Cell structure, about your lysosome, which is obviously a organelle. So yeah, it's really hard to know. Hey, I don't know if you remember, but I got my cellular control and patterns of inheritance. That's amazing, Kimmy. That's awesome. 89%. Wow. That's really, really amazing. Now, interesting, if you were at the school that I worked at before, that would be an A and not an A star. They used to make A stars 90%, A was 80%, down to 70, and so forth. And I would always say to the students, right, this is what the school say you would get, but actually in the real exam, that would be an A star. So that's awesome. That is really, really good. Congratulations. Now, luckily, biology is not the same as chemistry, because chemistry, you need 90-odd%. And this is the thing, Kimmy, I love that. I absolutely love that, because this is the point in the year for majority of students where there's like a light bulb moment, and it's a case of you're like, oh, that now makes sense. Oh, that is why I didn't get it before, because it was like a jigsaw puzzle where all the pieces were some up, some down. They were all over the place and they weren't connected. And once you've got that connection, it's literally like you're like, oh, I get it now. We don't have, no, you don't have A stars. If you're year 12, they shouldn't be predicting you A stars unless you are getting like 95% in your AS, because the highest level you can get in AS is an A. Hi, Carol. Carol, good luck tomorrow. I'm going to text you later. But if I forget and you're still on here, good luck tomorrow. I love you. It's my sister. We don't have A stars. I've already read that message. Right. It is getting time for me to think about going to bed because I'm really tired. I know I got very excited there. But I love it when it just clicks for students. I love it when students actually get the content and understand it. Because I remember being that student that hated it. I remember being the student who just thought I was dumb and everybody else around me was really smart and really able to succeed, and I wasn't. I was just a failure. And it was never that. It was that I didn't understand how my brain worked. It was that I didn't believe in myself. My self-concept was basically in the bin. And so now I know that, one, believing that you can do it is half the battle. Knowing that it's going to be a challenge is okay. Trying your best is important. But if you go into the exam and you're like, I'm not going to pass, so what's the point? You're automatically setting yourself up for failure. So you need to go into every exam knowing that it will be a challenge, but you're going to try your best, and you're automatically getting your brain engaged. The other thing is, if you haven't done it already, is doing the Superman pose. There's a thing about basically power posing, sitting upright and being like, but not really doing that. But what you do is you lengthen your spinal cord, and that therefore starts connecting your neural pathways, and that makes it easier for the impulses to get through and for you to then engage your brain, supposedly. But I believe it. I believe that if you are able to go into an exam and know that it will be hard, it will be challenging, it's meant to be. It's not meant to be easy. And some people will come out and be like, oh, that was so easy. And it may be easy for them, but actually for the majority of people, it's not easy. And some of those people who come out and say it is easy haven't read the question, didn't understand what was being asked of them, And that's why they found it easy, because they were like, oh, I could write loads and I knew what I was talking about. And then they're shocked when results day comes around and they're like, oh, I didn't do so well. And that's because they didn't read the question, or they misinterpreted the question, or they didn't take the time. So if a student comes out of an exam saying to me, oh my God, that was amazing, I literally am like, oh, and that tells me they've not had a good exam. If they come out saying, yeah, it was all right, there were some hard questions, there were some questions that I was able to do, then absolutely that's fine. But if you come out an exam saying it was easy, you haven't read the question. There's no way you are getting those high grades, because you should find it challenging. Most teachers will find it challenging. Anyone who is saying they don't find these exam papers challenging, it's not true. As an examiner, it sometimes takes me a number of times to be like double checking. Can I give that? What are you saying? Am I going to actually give you those marking points? And so therefore, it is we get trained on what we're allowed to accept so that we know whether or not we can accept your answer. It's not an easy exam, and it shouldn't be, because it's a level 3 qualification. It's not something you walk out with just by doing nothing. You should be working for it. But for some of you, I'm sorry, you have to work harder, just like I had to work harder. I work really hard on my study guides, my revision guides, and I'm currently, I'm very excited, I'm currently on the process of, it will hopefully within like a week and a half's time maybe be finished my module, almost full of module 2 now actually. So it's going to have biological molecules, biological membranes. I need to add in enzymes, also a bit of module 4 with the communicable diseases. So yeah, I'm excited to get that out and going. And then I'm excited to get the intensive course that I'm going to run. I'm going to run a full day course over 2 days, 2 separate dates, for students who are interested in intensive Easter revision. I'm going to run 2 days. I just need to finalize the dates, and there'll be 6 hours. And then for those of you who can't attend it, who won, I will also be doing a having the videos that you can have. On a random one, is cellulose flexible or inflexible? Because different years, mark schemes have said different things. It depends on the question. So cellulose is a molecule which is obviously going to have hydrogen bonds between, so cross links between the cellulose microfibrils. And when you are looking at the question, it depends on the flexibility of the cellulose. So if you were looking at, for example, auxin signals, it's a chemical messenger which signals for cell elongation, and for the cells to elongate, they need to have their cell walls be more flexible, and therefore the cellulose actually becomes flexible because of expansins, enzymes that are going to break the crosslinks between the cellulose microfibrils, and that allows for the flexibility. However, when you have the turgidity that you have, so once it gets to the point where it can't expand anymore, that is where it becomes inflexible. So it is a bit of both, but it really depends on the question that they've given you. So the mark scheme will be specific to that question that they've done it. Oh, hold on. There we go. So yeah. Right, I am going to go. It is 3 minutes past my bedtime and I am exhausted. Fingers crossed my eldest actually, does not wake up and make me sleep in her bed again tonight, because I really don't want to. Being in a loft bed, which is a single bed up on top of the room, is not much fun. So I'm hoping I get a full night's sleep in my own bed. And if I don't, I might not be as productive tomorrow. You are very welcome. And Darrow, thank you again for purchasing my revision guides. I hope you still find them useful. And yeah, thank you for watching my videos, and thank you for asking the questions.